Уважаемые коллеги, в рамках третьей лекции
Dear colleagues, within the frames of the third lecture of Metacomparatitivistics of MOZOLIN reading, we'll give the floor to one of the most well-known popularizers of science, fighter for academic scientific honesty, and first of all, a representative of natural scientists. Senior researcher, Institute for Information Transmission Problems, Kharkiv Institute, Russian Academy of Science, Doctor in Life Science, Panchin Aleksandr Yuryevich. Aleksandr Yuryevich is also a member of Commission on Russian Academy of Scientists to fight pseudoscience. Well, it's also important because I would like to kind of boast here and to advertise to colleagues protection from dark arts and some technologies related to the problems of genetics. And I would like to recommend to my colleagues, these are wonderful books, but I was really impressed with the humor in protection against dark arts. And we are really happy to see you and hear you. And of course, the name of your beautiful report, Playing God, has science crossed the line in genomic research? We would like to know whether it has crossed or not. You have the floor. Well, thank you very much. I will start my presentation. Well, I see you nod. Once again, hello. Thank you for having invited me to have the floor. And well, it interfered into genetics and into the life as it is. It is very popular among other things, and it has some negative impact, like Jurassic Park. Scientists create dinosaurs, and or in Black Mirror, they are trying to create robotized bees and the story about Frankenstein.
Что это франкенфуд, это еда, которая вас съест, да, что это что-то
And when we speak
апокалиптическое.
about genomic food, it's like frankenfood, something, some food with that will eat you. But I will not speak about GMO in some adverse effect. I will say a few words that the most of genetically modified objects is not the God play in this global sense. There's nothing global strategic happen there. All these yams contain these bacterial genes. It happens in nature that organisms mutate, or maybe some genes transfer from one organism to another, and there is no supernatural about it. If we want to prohibit and ban all the transgene food, then first we would have to ban potatoes, and they will be like that in prison. But I will not start with scientists and scholars playing with God, but saying how they outsmarted him. I think that's more interesting. But first, I would like to say why they need to play God. There's a great website, WTF Evolution. On this website, you can see great pictures, a lot of memes with animals like, why would you do that, evolution? WTF evolution. And then evolution tries to say that, well, we had difficult times, we had to scrape, we had to live on. But in general, yes, we do understand some animals have camouflage, some can mask, some can hide, but sometimes evolution made some weird things. And if you would design something that's just weird, you have this nervous laryngeus inferior nerve. It's for several nerves and several meters. You go down the throat and then you go up the neck. But you could have this nerve for only several centimeters. You don't understand why this happened. It happened during the evolution. Their ancestors did not have this long neck, but you need big changes, and big changes is not about evolution. Evolution is about small changes, small gradual changes over millions of years, and that's why giraffe would have this weird laryngeal nerve. Here's a table created by a professional designer. You can see that it's an ideal table, let's say. They do it whatever they want. And this is how evolution makes their tables. You just take tires, you put a tray on top of it, and you have an improvised table. It has some resemblance to the past, what it used to create this table from. But there's a problem with local maximum. Here's the picture to demonstrate it. Imagine that the most adaptable transportation means survive. During the evolution, we have motorbikes. Motorcycles are really great. They drive fast and they are great. They can transfer people. Hypothetically, automobiles or cars could exist, which would carry more people and have some more advantages. But the in-between forms are not really great. Going from motorcycle to a car, they would not survive. They would not have anyone behind. So as a result, you believe that a car is something that you cannot achieve. And then you need some kind of designer who would interfere and redesign it. In biology, genetic engineer could become this kind of designer, which will bring and narrow this gap. Craig Venter is a famous scientist. Even Newsweek said that they play God. Why did they say this? Because he, for the first time in history, created a bacteria, which was a synthetic chromosome. We compile a DNA, we have a ready chromosome and doubles, then it copies, and we have two cells. Each of them gets a copy of this chromosome. One bacteria is one circle chromosome usually. And what he did, well, basically you take any DNA and you copy it using special segments. He had a lab where they use a chemical approach to collect a letter. With the letter, they get fragments of DNA, they chain these fragments together, and they put it into the bacterial cell, where they take the natural chromosome first and they replace it with their artificial chromosome. Obviously, this artificial chromosome doesn't really change from natural. Oh, besides the fact that they put a watermark on this chromosome with the names of the scientists, maybe some golden discs with Justin Bieber inspirational quotes. But in general, you do understand that we can have a natural, or rather an artificial bacterial chromosome. But obviously, this was not the end of playing the god. We have principles for DNA propagation, for DNA copying. DNA has 4 letters, TAGC, and has a great principle for complementarity of letters. G in a double spiral or the double helix of DNA is always against T, T against T, T against A, G against C. And that makes it possible to copy itself. If we have T here, then it will have A opposite. Same applies to G, it will have C opposite. Thus you have double helix of DNA, because with some probability you might have mutations. And this is the foundation. We have DNA built the same principle. Humans, Aleksandr, Panchin, flies, mushrooms, flowers, and here scientists come and say, let's have an 8-letter DNA. This does not happen in nature, but they did it. They have 8 letters using the same complementarity principle. And what they did, they said that you can read these letters, you can read an mRNA, which assists in enzyme synthesis, and they code this green enzyme which shines, which emits light in darkness. So we do not just play God, we outsmart, we outplay the God, because, well, they did not do this. Here what we can do is a DNA origami. Obviously, you know what origami is. It turned out that we can use a PC, we can design some figurine, and then software would forecast what kind of molecules we would need to synthesize, so that then we will be able to have these figurines from DNA in the sedimentations, like we would have humans, cats, and smiles, and dogs. It seems abstract and funny, but if you can create a smile, you can create a box of DNA. And DNA boxes could have a lock, and you can put some kind of medication inside this locker, or maybe inside this box. And this box would be open inside the target. And so basically you deliver a box into a disease, then it would open, and then it would spray the DNA inside of it. This could be used for medical purposes as well. As of now, we do not have some specific medications, but we do have researches, and soon we will have something great. Okay, let us outplay the god one more time. We have central tenet of molecular biology that we read RNA. RNA is a single cell molecule. It has 4 nucleotides. It has AUGC instead of ATGC, but it's not a principal point. They synthesize enzymes, and I would like to run a video for amino acids formation. This video, I hope it runs well, how it happens inside the cell. Can you read it? Can you see it? Okay. The red is the DNA, blue is an enzyme, which is called RNA polymerase. It creates a polymer out of an RNA, and yellow is the RNA which synthesizes. You can see it inside the nucleus happening. And this is the speed at which it happens in a real cell. Then RNA goes out of the nucleus through the pores, and it happens to be in the cytoplasm of a cell, where this RNA molecule will have a ribosome, a special element which puts through this RNA through itself, 3 letters at a time, and each 3 letters correspond to 1 amino acids. It has messenger RNA, you can see them floating around, and messenger RNA has amino acid on the one hand. These are that amino acid which is used in enzymes. On the other hand, they have 3 RNAs, R against T, G against C, and so on. They have complementarity principle, and they move these letters from one letter to another. That's why mRNA has 3 letters which transfer it. And this point here, why it's great? Well, because the principles for these 3 letters of nucleotides to be coded by amino acids is universal. It's the same for me, for plants, for mushrooms, for typical mushrooms rather. Basically it's a universal genetic code. On the left, you see a DNA molecule, and you see 3 letters of nucleotides. How do you understand which amino acid will be coded? On the right, you can see the diagram for AUG, and you can see this specific code for AUG. You take the first letter, A, we go from the center. AUG will be methionine. The first codon will code methionine, second will code threonine, and so on. Basically, the whole life has universal genetic code. During the whole evolution of life, this principle stayed the same. But what's interesting here, you can change it. There are some exceptions in nature, actually. It actually changed a little bit, but why it did not change for millions of billions of years, why it stayed the same for billions of years. Suppose we change a codon, like ACG would not code serine, but lysine instead. What would be the result? Normally, a human person, any normal organism has thousands of genes which code enzymes, and suddenly all these enzymes change one of the amino acids. And I said that evolution goes in small steps gradually, but when you break thousands of different enzymes, you will have something bad, really bad. The organisms will be dead, basically. That's why these kind of mutations are not possible under normal condition or are very unlikely. Here we have George Church, and you obviously can see the play words. His surname is Church. Really, he has to play and outsmart the god. And he said we could have a beautiful solution. We can edit this genome. We can take a cell or a bacteria. We can get all this ATCG, and you can see that ACC, it codes different letters. ACU, ACC, ACA, ACG. Let us code all these 3rd letters into ACA. Obviously, they will have the threonine, but ACC is not used anywhere. Then we can rewrite and code with ACG some other amino acids. This is what they did. And the last work was dedicated to the ambitious idea to change 7 codons 7 three-letter couples, three-letter combinations. Scientists can change things which stay the same for billions of years of evolution, and that's astonishing. That's great. I already mentioned CRISPR, Cas9 mediated gene editing. We can change genetic material in different organisms. One of the most popular, obviously, is CRISPR-Cas9. We had He Jiankui, a Chinese scientist, who said that they got genetically modified twins who wanted to improve HIV immunity for these twins. We have AIDS, which gets inside the body and starts to act with CCR5. But there are some people with mutations, in the good sense of this word. They lack some letters in the code, and because of these receptors, they do not connect with HIV, and that's why they have HIV immunity. There was an interesting topic about a Berlin patient, Timothy Ray Brown. He was sick with leukemia and HIV at the same time. For leukemia, he had to undergo chemotherapy, and he had to have a bone marrow transplantation. And he had bone marrow transplantation operation, but he was cured of HIV because the person who he got bone marrow from had immunity towards HIV. And that's one of the cases which shows that this could happen. And Chinese scientists believe that they could change this situation. They could have artificial insemination, and they could improve immunity towards HIV for the twins. There was no medical reason to do so. That's why he was criticized, because it's just easier to have in vitro fertilization, and they will not suffer from HIV. You do not need to improve their immunity to HIV. Everything will be fine. But he did that. This is what happened. We're not sure if they succeeded or not, but this was the first case. Well, at least I believe that they tried to do so. They succeeded at least partially, but there was no publication on the topic. He used CRISPR-Cas9. I will try to explain what exactly that is. It's a popular technique. What's the origin? Usually, gene editing takes technology from nature. Nature invented something like called, let us call it, well, bacteria have something similar to computer antivirus. You have a bacterial cell at the top, but we have a bacteriophage which looks for this cell. And this could result to some diseases, and they could die. But sometimes a part of this bacteria inserts inside its own genome a special element, which are called CRISPR repetitions. And you can see this CRISPR repeats. It's like a database. It's incomplete code. Well, bacteria faces different elements, different diseases, different viruses, and then it puts this information inside this database. What happens later? It reads RNA, the sequence is similar to viruses, and then you cut it, and then Cas9, basically it's a biological scissors, you take this RNA molecule, and by using the complementary principle, which I mentioned already, G versus C, A versus T, they look for similar elements of DNA, they find it, they cut it, and substitute it. So this bacteria could fight viruses in future. And scientists said, well, you can use different RNAs, you can program it, you can use the scissors for different purposes. As a result, there are multiple ideas. Suppose we want to improve immunity of our plants to a specific virus. We can graft a Cas9 gene, or maybe we could have this directing, guiding mRNA for this virus. Cas9 will cut it and will improve its immunity. Maybe we have a human with HIV cells. HIV can mutate inside the cells. We can get this enzyme which codes HIV, and we can cut out HIV cells from human genome. So this is a possible medication for HIV, which we might see soon enough. But we also could insert something into the DNA, and we can cut it out and then insert something else instead of it. I will tell about it later, but first, I will speak about CRISPR-9, another report about mutagenic chain reaction. It sounds maybe a little bit scary, but we have physics law, which are fundamental, but we have biological laws like the Mendel laws on hereditary. And it turns out you can bypass these hereditary laws. Suppose we have a human person, they have two chromosomes, two copies of each of these chromosomes, and we have a mutation on one of the copies and there is no mutation on the other. If I have a child, it will have a 50% of getting the mutation, 50% of not getting the mutation. We will have the classical Mendel case for the attributes inheritance, for the characteristics inheritance, but we can get this green mutant, we can match it with a usual fly, and all their children will be mutants all the time. It turns out that it will be very easy and fast to propagate the mutation. How does this happen? This is a very complicated issue, but it's a very interesting one. We take an embryo cell of this fly and we put an interesting genetic structure there, which has Cas9 and gRNA, guiding RNA, for some specific place inside the genome of that cell, which we want to modify, where we'll get an organism from later. Cas9 enzyme synthesizes, it gets the place where it should go to at the picture A, and it cuts it. But what's important here is that these genes, Cas9 and gRNA, are between DNA elements which are similar to what is to the sides of the cut. Why is it important? When a cell finds that there is some kind of cut inside the DNA, they want to narrow the gap, they want to sew it. But if they do it, Cas9 will cut it again. The cell will narrow the gap, Cas9 will cut it again. But they can go the other way. They suppose, let's say, quote unquote, the cell supposes that something might have been inside or maybe between these two elements. How do we do this? Maybe they skipped it. If they skipped something, then we can use one chromosome and build the other chromosome in its image. Sometimes cells fix themselves by doing so, but they have two copies. Obviously, they copy and there is nothing wrong, so they can just narrow the gap. But because we took Cas9 and gRNA, only because we have these side elements, the cell can confuse our construction with the second cell. And if we do so, then it will take a different, it will use our Cas9 gRNA instead of the second copy. And once it does it, once it passes to the next generation, they will have mutant and non-mutant chromosomes, and we will pass this mutation to the next generations. If we place an additional gene between these red elements, then all the children of these mutants will, suppose, be able to light up green in dark. Or maybe we could cure HIV. How could we use it? It's a video from Australia. A lady tries to save herself from crazy rabbits which want to, let's say, attack her. Australia suffered from overpopulation of rabbits. Rabbits endanger different systems, but they have great superpower. They are very cute. Obviously, you cannot kill all the rabbits. They are super cute. So what might you do? suppose we could use a mutagenic chain reaction. We could have mutants, male rabbits, and all their following generations would be males as well. They would match, and there will be fewer and fewer females, and the general population would decrease drastically. Nobody actually proposed the same project for rabbits, but in New Zealand, they actually had this kind of project for other animals. They want to implement it by 2060, but it's actually very difficult to do so. They use genetic engineering for insects. For example, in Florida, there are Zika carriers insects. For most people, it's not dangerous, but for pregnant women, it's very problematic. And they started to fight these mosquitoes. So why and how do you fight mosquitoes? Suppose we could have artificially created and artificially modified, genetically modified mosquitoes, which will not have any following generations. Suppose we let them out in the wild and all the mosquitoes would die. But there is a problem. If there is no generation for these animals, how do we have enough number of these mosquitoes? But their larvae live in water. They need special antibiotics to live. We can get any number of mosquitoes in laboratory and then let them out in the wild. They will match with natural mosquitoes, and then the whole population would be extinct. Obviously, we will affect the natural system. We will need to get rid of the Zika mosquitoes and then let out normal mosquitoes. But when people heard about that, these are genetically modified mosquitoes. What if I'm bit by, what if genetically mosquito bites me? But obviously we're working only with male mosquitoes, and males do not bite. And even if they bite you, it doesn't really work like this. Maybe they watched too many Spider-Man movies where a radioactive spider bites Peter Parker and they turn into a superhero. But this doesn't really work. Scientists actually created a cow spider or spider cow. They have web in their milk. Their milk has mutations, and spider web is very durable. They use it for Kevlar protection, and one American company decided to match and to mix these two animals, cows and spiders. And you can basically milk this cow for web and then use this web for Kevlar protection. Speaking about superheroes, we have Incredible Hulk, who gets really pumped up and really strong when they are angry. We have this overpowered mice. Myostatin improves, or rather stops, muscle growth. If we break myostatin, then we can get overpowered mice. We can get some other enzymes in mammals, and they would be light, they will be green in ultraviolet. So Incredible Hulk is green all the time, but genetically modified Hulk will be green only during the night disco, or maybe then they go to nightclubs. But we actually have these kind of cows. They are over muscled. You can see this oxen. They say this is GMO, but actually no, it's not GMO at all. It's natural selection, but they have the same mutation. You can see that top right, we have natural cow, top or rather bottom left, we have GMO. So there is no reason. If this mutation happened in nature, in laboratory, by selectioners, it's a useless topic to discuss about it. When I speak about GMO regulation, it's all about politics and rhetoric in terms of biology. GMO must be regulated like selection products and the rest. Another case from superhero history, can you become a Cyclops like Scott from X-Men? It's a question actually for physicists, but we do have Cyclops in nature. This is one-eyed ship, and you might say, oh, there's some kind of ship from Chernobyl, or maybe some scientists decided to play God. But what actually happened? Her mothership was pregnant with her, and it ate this bioeco, absolutely natural grass, which has clopamine inside of it. Clopamine does not induce mutations, but it blocks Sonic Hedgehog gene. And this is the result of this development. Why do we know this? Well, because we can do this in laboratory. We can take a mouse, we can break Sonic Hedgehog gene, and we will see that we'll have one eye develop instead of two eyes. We will get a cyclops instead of two-eyed mice. The same happens in humans. The point is, it's all about consequences, but not the way we got them. If something is bio and eco, doesn't mean it's good for health. I spoke about genetic methodologies which you could use at first onset stages, but we could have an adult. Say they have a genetic mutation, how can we help them? They have trillions of cells, and it's really problematic to change something inside of each of the cells. And here we have gene therapy. We learned how to steal an ability from virus to get inside the cells of humans and get these materials. For example, we have nanoviruses, which adenoviruses rather, which can get inside the cells. And Sputnik 5 is all due to adenoviruses. How it works and why do we need this?
Well, let's imagine that there is a person who has a hemophilia disease, and this factor of blood problems with blood, you can only change the DNA in the cells of liver, and that is why we take a vector with gene coding factor, and then it delivers namely to liver, and we get that a person is diseased from a disease which was incurable before. He suffered from hemophilia. Nothing helped the Prince Alexei at the beginning of 20th century well, and this change will not be inherited by the next generation, and this genetic change does not change DNA of a human being. It adds another gene which will be produced by these cells. Small details. Another example, epidermis disease, which is easily infected and it really hurts. It can result in some early sepsis. Here is a child with disordered skin. They take his skin and grow layers, and later then do genetic modifications correcting the genes which result in this defect, and they get skin of this patient in vitro, and then they transmit skin, and now the child can enjoy life. With the help of genetic engineering, we can fight some diseases, especially cancer kind of diseases. So we take cells of immune system which could identify cancer cells. So they actively fight cancer permanently in our body. We do not even notice this. But if it were not for immune system, we would have been much more frequently sick. And we see that the cells of immune system and they identifying cancer cells in vitro, we add a specific receptor to detect cancer genes. It is already being used, genetic therapy for leukemia treatment. Let's also talk about other technologies, not only about genetic engineering. Elon Musk, for instance, offered such a thing which is called Neuralink. We can implant a device in brain which would read activity of brain and with Wi-Fi send it to computer. This story is not that far from reality. Here is a monkey, and they had they were paralyzed below the mid of the back. They couldn't use their rare legs. There was an implant in their brain which used wireless information transfer to send it to electrodes activating neuron cells, and they knew how to walk. But even healthy body can have use from this. You see that this monkey is controlling robotized hand, and she's doing it with the device which is implanted in its brain. Initially, it managed both its own hand and robotized hand. The brain get adapted to this, and if a monkey can do this, a human being can do this as well. So can you imagine in the future we will have two hands, two legs, and also auxiliary bodies? There is optogenetics. Uh, Algae have it some kind of protein channel rhodopsin. This is in the cells of algae. And with the blue color, it opens blue light. It opens, letting positively charged ions, and it would allow its activation. But the scientists decided that it can be transmitted, and thus we would get the activation. We can see what it results in and see the how nervous system works. And as a result, there were totally different methods of curing different kind of diseases. There is a disease which will have a distortion of optic nerves, and it can be given to the cells, and then they transmit information to central nervous system, and then these cells start perceiving light even though they do not normally perceive it. It was trained at rats, and they can be cured from blindness. Another thing was done by Susumu Tanagawa, scientist who managed to edit memory of a rat. What was done? They take rats, specifically genetically modified rats, and if they are given if they are not given some medicines, then the nervous cells that are active produce neurotoxin and become light activated. Normally, they have medicines, and normally they do not have these cells in brains. For instance, we have triangle cell indicated in red, and until they are active and stay in this cell, the neurons that are active become marked. After this, we can put this red in another cell and introduce the current attack and activating the cells which were in the red spot. And suddenly this red is afraid after this moment, as if it was deceived, as if it was beaten in another cell, and they're not in another cage. So memory is not a property of soul. We can change it. There were nicer works, some memories about a good night with a partner for rats, naturally. And these reduced their stress when they could see blue indication to the brain. I would like to briefly talk about cloning. People imagine cloning. They grow kind of ready people in tanks. It's arranged like this. A person is born as a small child, then grows up with new memories. Actually, clones exist among us already. These are twins. From the point of view of biology, clone is a biological identical body, but we remember about Dolly sheep. What happened to their sheep, which are donors of an ovula cell? They take the cell with an empty eggshell and the genetic modified material. The original is borrowed adult cells from a breast. They removed the a nucleus, and then we get Dolly. And actually, the Dolly was not the first cloned organism. We also have in Russia, for instance, the mouse Mashka was cloned. But to be honest, they took a nucleus not from adult body, but from embryo, from another embryo. But actually, the same procedure of nuclear transmission. I'll get back to the story because now we know how to clone mice, cats, even monkeys. So there is a question when we clone a human being, and if we consider that this is just transition of nuclear, we're very close to this, and actually, people are almost cloned. We are very close to this because there is such a technology of children from three parents. When we use the technology of transition of cell from nuclear from one cell to another one, DNA can be in a nuclear, and mitochondria are dealing with breathing of cells, and they have a little bit different genetic code. You remember I talked about something unchanged, and they have some differences in genetic code, and there are genetic diseases, mitochondria, which are inherited by female line. A mother has such a disease, and she doesn't want this disease to be with her children, and it will be at regular process of fertilization. So mother A and mother B make a child, and with the bad mitochondria, they take the nucleus and transmit to mama C mitochondria with good mitochondria, and they bring it back to mother A. There are not so many genes in mitochondria. That is why it's not a child from three parents, but from 2 point something parents. But it's not the main idea. They use the technology of cloning, and we are very close to using this in order to clone a human being. Here is a girl which was born with this technology, normal, healthy children. And here, finalizing the theme of cloning, why can we possibly need this? We can save the species dying out. Here are mouflons which are in a red book, and for their saving, we use their relatives, cattle. We can borrow the nuclear from wild mouflons, add it to surrogate bodies, and grow them, and then reproduce the in Jurassic Park, there was also this thing, the resurrection of died out species with dinosaurs. It's not that good because we have not preserved the DNA of dinosaur as it was shown in the movie. We do not have genetic material. We cannot save. But the same person who wants to play God, he suggests that we can resurrect mammoth. If you had a frozen cell of mammoth, we could borrow nuclears, then add it to the egg and of an elephant and grow. But we have genome of mammoth, which is totally calculated. We know genetic difference of mammoth and elephant. We have technology of genetic editing. We can take a cell of an elephant, then mutation after mutation, we can turn it into the mammoth a cell, and then add it to the egg of an elephant, and then we'll see small mammoth being born by an elephant. Hypothetically, we can recover died types that were died out, and we can clone a human being, and soon we'll be able to clone our predecessors using this technology. And sometimes a girl will be able to get a small predecessor. uh, Hypothetically, it's possible. It's not done yet, but this is not supernatural. This is possible. And the last thing, another video. This girl, girl, they are dealing with prosthesis and prosthetics, and if there is some disease, they can pray, and disease will be cured. And unfortunately, normally the diseases are cured, those they can come just like that. And something as amputated a leg is not controlled by God, but technologists help these people. And here, a lady lost the leg at a terroristic attack at Boston Marathon, and she lost her leg. She was afraid that she will never be able to dance again, but she got artificial prosthesis. And here, for the first time after this, she is dancing in front of the audience. I don't know whether you can hear music. Maybe you cannot hear, and I'm not sure that music is given here, but you can see that the processes that are done by modern science are really incredible, and they are much better than if we turned to supernatural powers.
Ой, прошу прощения. Вот, ну,
I beg your pardon. So you understand the idea, and you can see that he has both legs with prosthesis. He also helped himself. So, roughly speaking, when I'm asked, should we play God? Can we play God? I say yes, we can, and yes, we must, because nobody else can do this. Thank you for your attention. Thank you very much, Aleksandr Yurievich. Of course, it's really impressive. And now we have to think about your speech to percept some discoveries that you have demonstrated. Of course, we knew something, but regarding prosthesis, well, I'm really looking through the sim, and it was absolutely incredible revelation to me. There is an old story, and since then it's even more progressive. I can hear it that they use a neuropulse, and it is almost perfect. I have a question like this, if I may. Of course, the audience isn't impressed if we consider a development of, let us say, this kind of serious development of genetics. This is like 7 years or 80 years, mid of 20th century. Well, depending on where you want to start from. And actually, beginning of genetics, this is the law of inheritance in Mendel. And if we're talking about DNA molecule structure discovery, then it is the mid of the previous, and mass reading of genome it's closer to our time. So not much time has passed. Not much time has passed, probably within the context of other scientists, and it's quite a young science, genetics, even though in it has demonstrated incredible uh, discoveries. And we say that we play God and whether we have crossed a border. This kind of tempo of development of technologies and the fact that everything works. You talk just like scientists, and I think it's absolutely fair. Well, we have recently talked to Vera Izhevskaya, and she was talking as a doctor geneticist. You are talking as a genetic scientist. I was also present as some scientist. I will not tell his name. He said that scientists can make any kind of experiments which will make it possible to develop science and to develop technologies. They will do it if there are no prohibitions indicated in new law. So, actually, we lawyers do not really understand what's happening. And considering the fact that the law can forecast development of events, it shouldn't be behind the technologies, it should go ahead of technologies. And even now, after 70 years, the development of this science, genetics, we make a question whether we play this or whether we exceed these limits. And what about 50 more years to pass? Don't you think that it's not society and not the state uh, that are ready for these discoveries? What do you think about it? Oh, well, I do not think that the society is not ready for that, because actually there is nothing so much dangerous for humanity, what I have mentioned in my lecture. Oh, well, okay, there are some things Well, scientists do not make any kind of experiments. Let's start with this. There are some ideas that we cannot make experiments on people without their agreement. If we interfere into the system, it should be done after thorough thought over. Well, any kind of non-prohibited experiments. So even when we talk about prohibited or not prohibited, they have some own corporate ethics. There are some things which are not prohibited, but which are not considered decent to be done. Well, the things that why the progress is important, why it shouldn't be stumbled, in my opinion, because there are problems that exist in society now. The problems of genetic diseases, problems of lack of food, the food production systems, saving of wildlife, and they can negatively tell on us. The story with coronavirus, this is an example of interference of people into wildlife. And in nature, there are lots of different viruses which people face. And actually, technologies are required, they're important, they solve specific problems. And all prohibition measures like GMO, they actually exaggerate risks. Some risks are really good, some mechanisms, and some risks are invented. If I eat genetically modified products, I will have some problems with my health. So it feels like when laws about genetic engineering in Russia were thought over, we have read lots of fantastics without consulting specialists. Yes, it's a very important thing, and we should necessarily get consultation of specialists and have objective and subjective answers. I agree with you in this respect. We to learn something else, and we should consider what genetics and biologists tell us. Thank you very much, Aleksandr. As far as I can understand, for now, science didn't cross this border in genome research. As you know, if we consider examples from my lecture, whether science plays God, well, I said we already overplayed God. We already do things which nature could not do. But it's not only in genetics. Cell phones are also incredible things. Yes. Well, basically, possibly, our Creator also gave us functions which help us to create other species and other objects. So thank you very much, Aleksandr Yurievich. This was Aleksandr Yurievich Panchin, Senior Researcher, Institute for Information Transmission Problems, Kharkiv Institute, Russian Academy of Science, Doctor in Life Science. Wonderful report, very impressive. Playing God, has science crossed the line in genomic research? Thank you very much. Thank you. Goodbye.